So today we will be focusing on the first workflow that Dora would be presenting, which will be building a drug discovery workflow in 8 steps. So Bilal has already given an introduction about that workflow yesterday. We will be today focusing on the workflow and how to build parts of it, because I don't think so that we will be able to build such a complicated workflow in 1.5 hours just, but we will be trying to explain the entire workflow and show you how you can build the parts of that workflow so that you can replicate the same with other parts also. Okay, and just like yesterday, what we would do is we would take a break somewhere around the 45 minutes to 1 hour time slot, and then we will again continue with another 30 minutes. And if you have any questions, you can put it directly in the chat, or you can unmute yourself or ask us. So we will just wait for that particular section that is being discussed to get over, and then we will take those questions. Okay, so over to you, Dora. I'll just give me a second. I'll make you the presenter. Yep. So over to you now, Dora.
Okay, thank you very much, and welcome everyone. Can you hear me well?
Yes,
okay.
I can hear you well.
Okay, and I guess you can also see my shared screen with the summary of the workflow that will our topic today. So this is what Bilal has presented yesterday as a summary. This drug discovery workflow in 8 steps. Here our goal is to search for and identify new compounds, new promising compounds which have the potential to be used as an inhibitor on a CNS-based target, namely the human dopamine D2 receptor. And in order to search and find such new compounds, we are going to use virtual compound library generation, also characterize the generated virtual compounds based on their physical chemical properties. Then we will also use similarity searching in order to find real accessible analogs or easily synthesizable analogs for these virtual compounds. And finally, we will also try to predict the biological activity of the compounds in order to be able to select those molecules which, besides their good physical chemical profile and easy accessibility, have also good potential to be active, to show activity on our selected biological target system. And now I will go to KNIME and continue with the hands-on part. So this is again the very same workflow that you have already seen, and in the first part, in the larger part of our session today, I would like to go step by step or meta node by meta node to cover the entire workflow and show you what are the key steps, how they are connected, how the data flow looks like within this workflow, and what kind of configuration options we can use by working with this chemical data. And finally, hopefully we will have some time also to do some modifications and extensions on this workflow together. So let's start. The beginning of this drug discovery workflow is
Dora, sorry to interrupt you. I had sent all of you the link for this workflow. I guess most of you would have downloaded it. So if you have any questions how to open that workflow in a KNIME or anything like that, if you would want, you can also keep the workflow open so that you can also try it out at your end. That is also feasible if you want to do that. So Dora would be explaining it. then you can also try it out at your end. So if you want to do that, you should have the workflow opened in KNIME right now. If you have any issues in doing that, please let us know. We will be able to help you in that also. Okay, sorry, Dora, you can continue now.
Thank you very much. That's a good point. Thank you, Sarojit. So one more thing that I have the largest part of the workflow executed already. However, you can also follow and run the workflow steps on your own computer, because I will also explain the key points and talk about what is happening. So the workflow is using a relatively small data set, and the steps should be finished in less than a minute, most of them. So it's possible to execute them live. Is there any question regarding opening the workflow, or do any of you have any issues with it?
I don't see any questions in the chat box, so I guess most of them could have opened it.
Okay, that's great. So let's start. The first step is, because in order to find such, or our quest for finding new promising biologically active compounds, it starts from a set of already known and already tested compounds. Some of them has showed larger activity against the dopamine target that we have selected. Some of them have less or smaller measured biological activity, but all of the input data, all compounds in our input data set has at one point been tested and assayed against the dopamine target, the D2 target, and we are going to use this previously tested compound set as a starting point to analyze first, and to serve as a basis to come up with new virtual compounds. So in order to, if we want to import a chemical data set, we can do this with this mol importer node here. This is one of the nodes in the JChem extension, the ChemAxon Infocom nodes. And it can handle, if we go to the configuration window of it, it can handle different chemical data files and formats. Basically, every file format or every chemical format that is supported by ChemAxon products can be imported with the mol importer node. This can be an SD file, as it will be in our case, but also SMILES or INCHI or MRV or chemical names can be also imported. And one thing that have to be specified is the location of the file, of course. You can use an absolute file path if you have the file, you have the SD file on your computer, so you can browse for it or specify the absolute file path to this input file. Or another option that I've used here is using a relative, a workflow relative file path. It's a quite handy solution if you want to share workflows with other users, because also what I did here is that I did the input files and the configuration files in a folder right next to the workflow itself, and by using the relative passes, I could easily refer to these files in the accompanying data folder, and this way you can use the configuration as it is, so you don't have to browse for the files in your own file system. If you are importing a file which contains other fields and other properties besides the structure itself, then you can also import them with the mole importer node. If you want to, right now we don't need that. We are only interested, or actually we will need that to be correct, because besides the chemical structures themselves, we also will import the p-Campbell value, which is the actual measured biological activity of these experimentally tested compounds. So it's quite important to add those p-Campbell values as well besides the structure, so you can specify how the node should, or the importer node should look for these extra properties, and what should be added to the imported dataset besides the structure. And one more thing is that in the output options, you can decide in which format, in which chemical format you want to have the imported molecules. The default is the MRV, and we will leave it like that, because with the MRV format, we can use all the chemical features in the molecules that can be present. So once we have that, we can run or execute the node. I've already done this before the workshop has started. So, however, it should finish quickly because if I open the output table, you see that it contains a bit more than 3,000 compounds. It's input SD file, so it's not a large file actually. And you can see that besides the Campbell ID and the mole weight and formula, we also have the p-Campbell field with the measured assay results. Okay, so we have now the input data set. Now the next step, as in any data analysis and data visualization task, is the cleaning and standardization, normalization of the input data. This is what we can do with this first meta node, the second step of our workflow. So it will be it will take care of the data and the structure standardization especially, and we will also have a duplicate filtering step. So I will open this meta node to see the details. And here, one of the key steps is the structure standardization, for which we can use the standardizer node, again from the JChem extension bundle. If we go in configuration mode, you can see here on the right the standardizer configuration that I'm using here. If you want to, you can really modify this configuration, remove steps, add some extra steps from the collection of available actions on the left. To define or to specify the standardizer configuration, a configuration XML file have been attached to the workflow group that you have downloaded. So you should be able to have the same standardization steps configured already. If you want to, you can add a new config file with the import button, or you can also use a URL or copy paste from the desktop standardizing application, let's say. One more thing is a smaller detail that we can decide if we want to append a new column with the standardized molecular structures, or if we want to replace the original input column. I will append a new one because it can be useful if we can compare the original and the standardized molecules. Okay, again, I have the output table ready. So And actually, this is the first structure column is where we can see the original, the imported molecules, and this is the one which contains standardized molecules, and there is also a summary about which of the configured standardizer actions were or could be executed on the molecules in each molecule in the set. The typical standardization steps that are used frequently is to aromatize, de-aromatize the molecules depending on the need, so to have a consistent representation for aromatic structures. Also removing smaller fragments or salts from the molecules is something that are frequently needed. Sometimes we have to neutralize our compounds or clean up some sterile features. Also, there can be a difference how certain functional groups are represented. For example, a nitro group is something which is known to have different representations, so it's crucial to standardize and have a single consistent representation in all of our data set. And also the hydrogen atoms in the molecule can either be represented as explicit atoms or implicit. So again, this is something where we have to standardize and consolidate our data set. Okay, so now we have the standardized molecules. In order to get rid of, to remove the columns that we won't need anymore, there is a column filtering step. So once we have executed this, we will only have the standardized molecules, their Campbell, original molecules Campbell ID, and the measured P Campbell value in the data set. And also we can add here, or we can check if there are any duplicates in our collection. It may happen that at the beginning we had some duplicates, or as a result of the standardization, we ended up with some duplicated chemical structures. So there can be several ways how we can do this duplicate filtering. The approach that I used here was to generate the SMILE string of the molecules using the advanced mole converter node of ChemAxon, and I added an extra column containing the generated SMILE string, and then I used the default KNIME node called duplicate row filtering to find and remove those rows where a duplication has been identified. And if I run this, okay. And we check that we have a bit smaller number of structures now in the deduplicated table than we had in the original one. So indeed, there were some repeating, some structures that happened more than once in the data set. So now we have a clean data collection. We have done with the input data pre-processing. So the next step would be what we would like to do is analyze this input data collection and see, and the reason and the objective of this analysis would be to find what are the typical scaffolds, or actually first to see if there are any typical frequently occurring scaffolds in this input data set, and whether those frequently occurring, or which one of these most popular frequently occurring scaffolds have the largest average activity, largest average peak Campbell value. Because if we do this analysis and identify such a scaffold, in the next step we can use it as a core structure in a virtual library enumeration process and generate a set of compounds by using this selected scaffold, which we can go to the next step and characterize it on their physicochemical properties and expected activity. So in order to see what kind of scaffolds we have in the input data set, we can use a hierarchical clustering based approach. This is what the next what is in the center of the next meta node. I will first start the execution of the meta node, and also while it's running, I will show you what are the details behind this hierarchical clustering and the following scaffold selection. So the key step or the key node here, which is being executed right now, is called library MCS. This is the node that can cluster hierarchically the input molecules by systematically looking for maximum common substructures among the molecules in the input data set. As a result, we will have a hierarchy which can also be represented, for example, in a dendrogram, where at each level, where at the bottom level we will have the individual input molecules, while at the next level we will have the maximum common substructures which were identified among the input molecules, and the next level we will have a maximum common substructure of the previous one, and so on and so on. So the topmost level we will have what we can call scaffolds of the molecules which have been simplified enough or for our purpose to be used in an upcoming library enumeration process. So the library MCS have finished, and now you can see what is the result of the enumeration. So we have a bunch of data describing this hierarchical cluster structure, and also, so this is what you can see here in the hierarchy ID column. You can see here how these levels look like, and we also have some information how many members of each of these hierarchical clusters have, what are the parents of the compounds, what are the compounds above them, neighboring level, so on and so on. So we have a bit more complex data set as a result of running the library MCS node. So any other node that we have added to this meta node or wrapped in this meta node serves the purpose of getting out the scaffold information, and also get information about how many times each of these scaffolds were found in the input data set, and finally what was the average calculated average P Campbell value for each of these identified scaffolds. And as a final, so here, if I come to, I think it was this joiner node, where we can already see the topmost, the class or the scaffolds or maximum common substructures from the topmost level, and there is also an average P Campbell value calculated for them. And then we can use a sorting node, then where we can use the number or the number of the elements, number of the child structures belonging to each scaffold, and we can also, as a secondary sorting parameter, we can use the calculated average p-Campbell value, and then we can use a row filtering to select the best performing scaffold from our collection. And if we see the filtered data set and enlarge it a bit, then you can see that this was the scaffold structure which had the largest number of compounds, or the largest number of compounds where this scaffold structure was found. And here you can see the calculated mean p-Campbell value. So we selected this molecule to be used as a core structure for around which we can enumerate new virtual compounds. Okay, so now we have the core structure, we have the scaffold to continue with the library enumeration. However, we can collect a bit more data from our input compound collection, because we can also check how this selected scaffold is substituted, how it is typically functionalized in the input compound collection. This is what happens here in this metanode, the R group decomposition metanode. I will again start its execution and then go into details and open the metanode. So here, what happens here is that I use the R group decomposition node from the JChem bundle, and this R group decomposition node requires a scaffold for which we use the scaffold that we have just selected. And it will also require a set of compounds, in this case our input, standardized input compound collection. And the R group decomposition node will use the specified scaffold as a query, as a substructure query, and look for those compounds in this column which contain this selected scaffold. And in those compounds where the node could identify the scaffold, it will also identify the different functional groups, the different substituents, and the position of them on the scaffold structure. So if we take a look at the output window, then you can see that it's a whole R group table with quite a number of columns, because our selected scaffold has 14 positions where it can be functionalized. So the R group decomposition node checked all of these 14 positions and identified the different substituents on them. After that, we can find which of those 14 positions are the ones that are most frequently substituted with functional groups that are larger than a simple hydrogen atoms. And we can also check what are those typical functional groups in these popular positions. So if we take a look again, if we take a look on those 14 substituent or substitution positions, then what we found that there are those. There are quite a lot of them, which where almost every time just a hydrogen atom occurs, and there are these 7 ones where we have something different than simply hydrogens, and then we can also take a look at them and find the positions. For example, the position 14 is the one where we frequently have substitution, and also position 5 and position 1 are again popular functionalization locations. So based on this analysis, we can check where we want to expand our molecules with the following library enumeration. And we can also get some information about the functional groups that we can use, that have already been used or which have already worked in the past. So all of this information, these steps can actually be skipped, and we can use a different tactic for defining and designing our virtual compound library, or we can use a different approach. So if we move to the
Dora,
next, yeah.
sorry, can we just pause and ask if somebody has any questions? If anybody is having any problems to follow till now? If yes, please unmute yourself and you can ask the question. If not, then we will go ahead further with that. I guess later on we will also see how we can create one of these meta nodes. Okay, so I have not got any questions yet. Yeah, Dr. Pramod.
Hello, Pramod here. Yeah,
Yes.
so we are not able to follow like this when I'm importing the nodes, then it is initial steps. The first step, it is not executing. So what? So we have like downloaded this drug discovery workflow.knr file, and then we try to import this and try to run it, but we are not able to follow it up.
So you could import the workflow, right? Sorry, Dora, take it over.
I wanted to ask the same question.
Yeah, is it for some people it not work? Surya here.
Yeah, Dr. Surya, yeah.
Yeah, there's just to open the node and just follow the browse, and you can double click it, it work actually. Are
Yeah,
you
Dr.
able
Pramod,
to,
if you can tell me.
yeah, Pramod, are you able to open the node?
So I have saved the file in my local system, and then I try to import KNIME workflow, and then
Yes.
I selected the
First one.
and then from the source I have selected the file by browsing it, and when I'm opening it, it shows that some of the element already exists in the target destination. Okay, so I already tried earlier.
Yeah, probably you're trying to import the workflow again.
Okay, so but I have in the KNIME Explorer, I have local workspace where I have ChemAxon KNIME and then drug discovery workflow also.
Click on this.
Under that I have data and results.
So please double click on the KNIME Explorer where you see the drug discovery workflow, and then there will be another window opening where you could see all these workflows.
Yeah, okay, there the data and then result is there, and under result there is ChemAxon under drug discovery workflow.
Yeah, just double click on that file.
Double click on that.
Yeah, when I'm double clicking, it is showing the workflow. It is showing the workflow, but the first one, the mol importer, when I'm right clicking it, the execute is not there. The configuration is there, but the execute is not. So I supposed to execute it, right? So it is not running there.
So right now you are able to see the workflow. Now the question is regarding the mole importer execution, right? So Dora, can you just so he is not able to execute the mole exporter workflow. So you see that in the at the bottom part of the mole exporter there are 3 dot points. In our case, it is showing as green. Can you say which color you are seeing?
Here.
So
Yellow. Should be yellow.
yeah, so I am seeing the red color.
Oh, that means
Correct.
not yet
Okay.
loaded. That means not yet loaded.
Yes. So in
Okay.
that case,
Okay, so in the KNIME Explorer, where you have drug discovery workflow, you can open that block, and below the drug discovery workflow, do you also see these other folders like data, then
Data and
result?
result, yeah, correct.
Mhm,
Yeah, I'm able to see data and result.
mhm. Okay, so I think if under the mole importer node you see the red dot, then this means
Yeah.
that the node is not configured properly.
Okay.
So
So
you should go to the configure mode,
okay.
and one thing that is mandatory to have is the location of the input file.
Okay.
Do you have this file location specified?
Dora, can we just Dr. Pramod, will it be possible for you to share your screen so that we can quickly resolve this?
Yeah, yeah.
Okay, I'll just make you the presenter. Dora, is that fine?
Yes, sure.
Sorry for the interruption, but I guess it would be useful for them now. So, Dr. Pramod, you can share your screen. Yeah.
Yeah, so I have opened so
Okay.
configure and then browsing it.
Yes, you're right. Browse.
That's,
CNS active,
Yes.
right?
yes.
Yes, yes. Open.
And then apply.
Okay.
No, okay.
Okay.
Okay, do okay.
Licensing issue, I think.
That's
License.
a licensing issue.
Okay,
Yes.
so it is the license that you have, it is not correctly configured because you see that there is you are getting an error regarding the license.
License.
Yes.
So
Go to the yeah.
Can you go to
User.
the
Go to the
file?
user. Go to the user.
ChemAxon, right? So I have put this license.xml
No.
file here.
This is one of the licenses that you need. Can you?
No, other license. Other one. Dot file. Dot DAT file.
Okay, .dat file.
Yeah.
Yeah. So you will need to go to preferences within KNIME.
Go to 9.
Go to
Yes.
9. Yeah.
Go to 9 now. Go to 9. No, here. Bottom. No, you
The
are
KNIME window.
application. Yeah.
Yeah.
And then file,
File.
preference, and the
Uh,
9,
9. Go to 9.
KNIME.
and JChem. And
Yeah.
here you have that
Now,
one.
yeah.
Yeah. You had to place the wrong license over here. Now try to do that.
Now it will work. Yes. It's working.
Yeah, it is still processing, I guess. Yeah.
Yes.
Okay.
Now it
Okay,
green.
great.
Okay.
Okay. Yeah.
Okay,
So
great.
now it is
So
working, I guess.
I am making Dora the presenter again.
Thank you.
Yeah.
So anyone else has any other questions? I don't think so. Any other questions are there? So we will just get, we'll continue with this. Yeah, over to you again.
Okay, thanks. So the next step is using the selected scaffold and generate a library of virtual compounds around this selected scaffold. This is what happens in the next meta node. Again, I will start the execution and go to the details. So after the analysis of the selected core structures, there were 3 locations, 3 atoms on the scaffold, which were identified as typical points, typical locations of functionalizations. 2 of these points were 2 equivalent aromatic carbon atoms, while the third one was the free nitrogen in the piperazine ring of the scaffold, and one by one with library enumeration steps on the virtual reaction steps, we can substitute these locations, these atoms. So the first step in the virtual compound generation is about the substitution of the aromatic carbon atoms in the ortho positions relative to the piperazine ring. We open this meta node. Here you can see that I used 2 different reaction schemas to do this substitution. One of them was a nucleophilic aromatic substitution reaction, while for the other one I used a Friedel-Crafts type alkylation to substitute aromatic carbon atoms. And if we go one more level deeper, then you can see that first we used our scaffold as an input, and then we have to make a standardization step, because in order to be able to use this nucleophilic substitution reaction that was selected, the key note here is this bimolecular reactor ChemAxon node. If we check its configuration, here you can see an example reaction of how such a nucleophilic aromatic substitution type reaction would look like. So we want to substitute one of the aromatic carbon atoms, however, on the scaffold. However, in order to do that, first we have to transform these aromatic carbons in the ortho positions and add a chlorine atom or replace the hydrogen atom with a chlorine atom. This is why we needed this standardizer node here before the reactor node itself. And then we needed the right type of reactants, which we import with another mole importer node from another file, which can be found in the reactant subfolder of the workflow group. And finally, we also have to import the reaction schema that describes this nucleophilic aromatic substitution like or SN2-like transformation. This reaction schema can be found in the reactions subfolder of the data directory in the workflow group. And besides specifying the actual transformation, this reaction file can also contain information about so-called reaction rules that can make the basic transformation more chemically relevant. Because with these reaction rules, we can define reactivity rules, selectivity rules, and different exclude rules as well, which will give a better control over the transformation. So, for example, we don't have a reactivity rule now, but we will have one. We will have those with further reactions. With the reactivity rule, we can filter out those reactant compounds which would not undergo the selective chemical transformation in real life either. By the selectivity rule, we can select which part of our reactant molecule will be transformed if there are more atoms or more parts of the reactant molecule which can be transformed, and the selectivity rule will decide which of those locations will be the preferred one. And with exclude rules, we can give additional criteria whether a reaction can take place with certain reactants. And other options in the reactor node is we can decide if we want to run the node in or a reactor in a combinatorial mode, so react each reactant, each molecule from the first reactant set with each molecule from the second reactant set, or if we want to have a sequential enumeration instead, where only the first molecule from the first reactant set will only be reacted with the first molecule from the second reactant set. And we are interested in the product molecules that the reactor node can give us, but we can also get the reactions themselves, and also we can specify the ratio of our two reactants. We can decide what to do with unsuccessful reactions, and if we want to, we can also generate a synthesis code, which later on could be used to use as an information source of how the transformation went actually, or what were the parts that were transformed. Okay.
Surya, 1 minute. How many reactions? Currently, I think you have given 3 sample reactions. How many ready-made reactions you have already?
Oh, sorry, I didn't get the question.
So,
The reaction?
Yeah. How many sample reactions you have, or otherwise, how many MRV file you have? Currently, I think you have given only 3 MRV files. One is Friedel-Crafts acylation, other one is this nucleophilic substitution, one more.
Yes, so those 3 reactions that I'm used here come from the ChemAxon reaction library. This complete reaction library contains about more than 200 organic reactions.
Okay, thank you.
Okay, and as an output, as the first output of this reactor node, we get the products from the successful reactions, from the successful transformations. Yeah, I haven't showed you the imported reagents. It was only a very small set of reactants because we don't need a huge generated library here. And here are the transformed molecules, the product molecules that we wanted to get, and with some data cleaning and column filtering, we will get only the product molecules which we want to take to the next reaction step. Okay, so this was the nucleophilic substitution, one of the ways to functionalize the aromatic carbon atoms. We can use another reaction as well besides this first one to add other functional groups. For example, with the Friedel-Crafts substitution reaction, and well, the scheme is very similar to what we had with the other reaction. So again, we import the reactants which we want to use for the substitution reactions, and again we had to do some structure standardization on the scaffold molecule. Where I wanted it. So in order for the reaction to take place, we had to attach a bromine atom to the free nitrogen on the non-aromatic ring. Otherwise, the scaffold would have been excluded because of the free basic nitrogen atom. So this is why we needed a structure standardization step here, and then we could use those 2 reactants as input for the reactor node, again, bimolecular reactor node was needed here, and we specified now the Friedel-Crafts calculation reaction schema, where you can see an example reaction, and we can also check the reaction rules. So now we have a reactivity rule as well, because in order to get chemically relevant products only, we have to check whether the aromatic system, whether the aromatic system in the scaffold is activated enough to undergo such electrophilic substitution reaction. So this can be done with the reactivity rule, and also there is a selectivity rule regarding the imported reactant set, which site of these reactant molecules will be used. Another thing that we can add here, we can also specify further selectivity rules if we want to have directional rules which regarding the aromatic carbon atoms of our scaffold. So now we don't have such a selectivity rule, which means that the reactor node will consider each aromatic carbon atoms in our scaffold as equally likely positions for the substitutions. However, if we want to, we can add a new rule that would select, for example, the ortho carbon atoms, orthoaromatic carbon atoms for the substitution to take place. And after that, everything else is the same as in the first reaction step. So we again are interested in the product of a combinatorial transformation schema, and we are adding the two reactants in a 1:1 ratio. And once we got the primary products, with another standardization step, we could remove the bromine atom that have been attached as a necessity here, and with a column filtering node, we will get the cleaned up product data set right here. As you can see, with this reaction rule set, we didn't get only orthosubstituted molecules, but the meta and the para positions are also functionalized. If we will have some time, we can also check how further reaction rules can be added. Okay, and now we have the two product sets from the two substitution reaction schemas. So with a concatenation node, we can add them together, and the two products set together in a single output. And now this concatenated product collection will serve as the input of the final reaction schema, where we want to use an alkylation reaction to functionalize the amine nitrogen of the piperazine ring of our scaffold. Here the process is quite similar, only the reaction itself is different from the previous ones, and also there had to be some pre-processing. We had to do some pre-processing on the imported reactant data set with some property-based filtering and structure standardization. So finally, with cleaning up the final data and renaming the columns in an appropriate way, we will have a column with the new virtual compounds. We have these 280 virtual compounds which can cover the space around our scaffold molecule. Okay, so now we have these virtual molecules. Our next goal is to select those virtual compounds from this library which have the best physicochemical properties. And so how do we define what is this best or what is this fitting physicochemical profile? Well, we can use the CNS MPO score as a property to assess the profile of our molecules. So now if I open this CNS MPO metanode, first we will have to calculate those properties, those physical properties which the CNS MPO score calculation requires. But even before that, we generate the major tautomer for each of the input molecules. And after that can come the calculation of those 6 physicochemical properties from which the CNS MPO score can be generated. Namely, these 6 properties are the molecular weight, the log P, the log D at pH 7.4, the topological polar surface area, the number of the hydrogen bond donors, and the strongest basic pKa values. So one by one, we can calculate those using the matching calculator, ChemAxon calculator node. So for the molecular weight, it's simply selecting the mass property in the configuration window. As for the log P calculation, we can also decide which of the available log P calculation methods, ChemAxon log P calculation methods we want to use. Here, the consensus method was selected. For log D, again, we have to specify or use the matching log P calculation method, because log D values are calculated based on the calculated log P and pKa values. So we have to use the matching log P prediction method, and also we can select which pH values we want to see. Well, for our calculation, the 7.4 will be needed, but we can have other ones. And also we can use the default properties for the polar surface area and the hydrogen bond donor calculations as well. And finally, for the basic pKa, we can also go with the default configuration options in this case. And as a last step, we can just remove the extra columns that are not needed. And now we have the primary calculated properties. Now the next step is to generate the CNS score from those. And as yesterday Bilal has mentioned, for each of these, from each of these 6 properties, there is a scoring function or a transformation function that transform the predicted value to a score value between 0 and 1. This is what happens in these individual metanodes for each of the predicted properties. So if we check, for example, the first one, which generates the score from the calculated mole weight value, is we can see that first we use a rule-based role splitter node, where we separate those molecules where the calculated molecular weight is less than 360 or greater than 500 from the other molecules where molecular weight is between 360 and 500. And then for the first group, we use a rule engine node, where we say that if a molecular weight is for a molecule below 360, then it's the mole weight score contribution should be 1, while for those where the molecular weight is above 500, the score contribution should be 0. And as for the second group, between 360 and 500, we have a simple mathematical formula which transforms the molecular weight to a score value. A linear function is used actually to transform the molecular weight to a score contribution between 0 and 1. And then we concatenate the two result sets, and here in the last column we get the calculated contributions. And then the same logic, the same approach is used for the other 5 properties as well. So only the functions, the shape and parameters of the transformation functions are different for each of the properties. And once we have the 6 contributions calculated, we simply add them together to get the final MPO score value. What you can see here. So this way we get MPO scores which are between 0 and 6. And the higher this MPO score is, the more likely it is that our molecule will be able to cross the blood-brain barrier and get to our target, our receptor in the central nervous system. So the final step in this meta node is a row filter, where we are selecting those molecules where the calculated CNS MPO score is at least 5 or larger. And we started with 280 virtual compounds. Now, as a result of this filtering, only 24 of them survived and remained in the workflow.
Yeah,
Okay.
Dora, I'd just like to pause again and just ask if there are any questions. I don't think we will be able to take a break today if we have to show meta node creation. So is there any questions anybody has for this till now? Okay, I don't think so we have any questions, so we can carry forward.
Okay, thank you. So now we have the best virtual compounds, 24 of them. So now the next step with good physical properties, now the next step would be to go from this virtual compound space to the real compound space and find real, accessible, purchasable, or easily synthesizable molecules which are structurally similar to these most promising virtual compounds. We can do this by searching for real analogs in different databases. One of those databases where we can find a large pool of easily accessible compounds is the enamine real database. So what the next metanode does is that it performs a large-scale similarity search in a subset of the enamine real database, and in order to find those enamine compounds which are structurally most similar to our narrowed down virtual compound library. Here we are not using a dedicated ChemAxon node to do this search. Instead, we are using the native RESTful nodes, because in order to do this large-scale similarity search, we are using the MedFast search tool of ChemAxon, which is a server-based application and which has a RESTful API. So with post and get requests with RESTful web service calls, we can communicate with the MedFast server and run and use it for similarity searching. So what you can see here, I would not go into the details of data processing here. Most of the things that you see here is just simple data manipulation to prepare first the post request, and here's the key node that sends these web service calls to the MedFast server, and specifying that we are looking for the most similar compounds, and compared to our input molecules. Then we have to process the answer or that we get from the MATFAST server. We get a JSON output first, which have to be processed. We have to get out the internal identifiers of the enamine compounds that MATFAST found for us. And as a next web service call with this GET request node, we can again go to MATFAST and ask for the SMILES strings of these enamine compounds that were found for us. So if we take a look at the output of the meta node, then we will have a bit more than 100 real analog compounds, because for each virtual compound we were asking MATFAST to find us the 20 most similar enamine analogs, and after we did some duplicate filtering, we ended up with these 113 real enamine analog molecules. And since they are structurally similar to our best performing virtual compounds, we can expect them to be good candidates to continue our workflow. However, to make sure that we only keep the best enamine molecules, we can run on them a CNS MPO based filtering again. Here, the process is the very same that we used previously for the virtual compounds. So again, we calculated the same 6 properties and transform them into the MPO score and use this MPO score for filtering to keep only those enamine molecules that have the best physicochemical profile. So now we have real molecules with good predicted properties. The only thing that is left is to get some kind of prediction for the biological activity that we can expect from these enamine molecules. We can use for this some kind of machine learning based approach. What was done here is just a quite simple and basic and fast machine learning step, where we took the original input data set and used it with a random forest algorithm to build a binary classification model which could categorize the molecules as active or inactive against our selected dopamine D2 receptor. So if we take a look at the next meta node, the first step is building this binary classification model. We can also visualize the p-Campbell values of our input data set in order to decide where how we should select a threshold that divides active and inactive compounds. So here you can see this distribution of p-Campbell values. For this task, the p-Campbell value of 7 was selected to distinguish active and inactive compounds from each other. And then we calculated different fingerprints for the input molecules. The chemical hashed fingerprint was one of them, and the ECFP fingerprint was another one. And finally, ECFP was used to build the model and then to predict the activity class of the new molecules. And then came the random forest learner node, which built for us the classification model, and also we used the test set of the test part of the input molecules and added a scorer node to get some information about the model performance. This is where you can see false negatives and or molecules predicted active or non-active against the labels that were assigned to them based on their P Campbell value. And there are also some activity or accuracy statistics available as a second output. So here you can see the overall accuracy for the built model, which is a bit below 85%. So it is not a really good model. However, here it wasn't the goal to optimize every parameter to get a really good performing model, just to give an idea how we can extend the workflow with some activity prediction. Okay, so we had the model built from the input data. The only thing left is using it to predict the activity category of the selected enamine compounds. For this, again, we had to generate the ECFP fingerprint-based representation of the enamine molecules and use the random forest predictor node to assign the classes to them, and then we can use a native KNIME node for assigning different colors to molecules which are predicted active or non-active, and clean up the data set again with removing the columns that are not needed. And finally, we can either check the output table and select the rows with the best performing, the most promising molecules manually or with some table manipulation, row filtering, or we can also do some visual analysis and selection using one of the interactive KNIME nodes. In this case, a component was built, a visual component was built, which contains a scatter plot of the molecules, which you can see here, and contains another panel where we can see molecules that we have selected. So in the scatter plot part, we can see the compounds, the enamine compounds, and on the y-axis are the CNS MPO scores. The red dots are for the molecules which were predicted to be non-active, while the green ones are the ones which were predicted to be active. And if we click on the non-active label, then we can remove the red dots and see that we only have, in this case, 2 molecules that were predicted to be active. We can also use selection or area selection and specify that we are interested in those molecules where the MPO score is at least 5.5. And if we make a selection, we get the structure of this single matching molecule. Now, if we want to apply this selection in on the output table, then we can use the close and apply option. And once the node has finished running, then we can also check the output table. So here we can also see the color coding with the many red ones and a few green ones. Or if we use a row filter node, then we can apply our previous selection on the output of the next node as well. So as a result, we will have a single compound that matches all of our criteria regarding physical properties, predicted activity, and easy accessibility. And if we want to, we can finish this workflow by exporting these remaining selected compounds to be sent over to synthetic chemist colleagues, for example, or others who also participate in our project. We can use for this the mole exporter node, where again we have to specify the location of our exported file, and we can also decide whether we want to add, beside the structure, any additional fields from our table. And this concludes the workflow itself. Are there any questions in this latter part or anything else that was mentioned?
I do not see any questions in the chat box. If anybody has any questions, please let us know so that we can answer them. And what I'll also do is I will send you a link in the chat box. If you want to use any of the other reactions that are there in the ChemAxon library, I have sent you a link. You can download the entire library and you can use them. You'll have to configure them properly. Apart from that, anyone has any questions? I don't think so. So another thing that I wanted to mention is that in this case, we are using the Enumeration real database using the MATFA similarity search. But in the ChemAxon nodes that you have downloaded, you will see that you do have JChem base as well as JChem cartridge nodes, which can be used if you are storing your data in a JChem table, and you can search against that database also. I don't think there is any questions, Dora. So I guess we can go ahead and like just create one of the meta nodes, if that would be feasible.
Yes, we can. However, another thing that I had in mind is that we can try to modify one of the existing meta nodes.
Okay.
So what I thought we can take a look at is modifying how or extending the virtual compound library generation, because yeah, we have a bit less than 10 minutes, so I think this could still fit in the time that we have. So one thing is that for the library enumeration, our goal was to generate some substituted versions of our selected scaffold. However, our goal was to generate ortho-substituted compounds. And once we used the Friedel-Crafts alkylation transformation, we saw that with the existing reaction rules, we didn't get only the ortho substituents, but some other compounds as well. So and also we only we only have mono-substituted compounds. So we can extend this enumeration step and virtual reaction step to have a better version of our virtual compound library. So first, if we go to the Friedel-Crafts alkylation meta node and take a look at the bimolecular reactor node and its configuration, in the rules window, there is where we can modify the reaction rules, reactivity, selectivity, and exclude rules that we have. And now among the selectivity rules, there is a condition or criteria only for the second reactant, so for the substituent, and there is nothing that would specify where the aromatic ring should be substituted. So this is the first thing where I would add some modification and would add another reaction rule. So if I want to select that aromatic carbon, which is the most favorable for this transformation, then I have to specify a chemical terms-based condition, which would select that carbon atom from the scaffold where the lowest where the adduct, the reactant adduct would have the lowest energy, let's say. So if I add an additional condition here, I can make this extension and make this reaction more chemically significant, and I can also extend the tolerance list as well, and this way I can specify which molecule, which locations of my aromatic compounds I want to allow to be substituted. Also, if I want to, I will not do this, but further conditions can be added or modified regarding the exclude rules as well. So if I apply these changes and run the reaction, okay, then I okay, I may have some typo.
Specificity, there is no
Yeah.
bracket.
Yep. Like this.
Selectivity. Yes.
Yeah. Better. So now the difference is that I have only ortho-substituted compounds and nothing else in the non-reactive part. So this is one way to make our reactions more chemically restrictive and more chemically relevant, let's say. Another thing is then how can we create reaction or products actually, and how can we where not only monosubstituted molecules are present, we can do this by adding a further reaction step. I'm just checking the clock if we have still some time for that. So Richard, do you think that we have some time for that one
Yeah, I
as
guess
well?
we can extend for 5 minutes. I think that should be feasible.
Okay. So if you want to add additional reaction steps, then we can go to the ChemAxon and the JChem node collection specifically and select the bimolecular reactor node and just add it to here, where in this second reactor node we will use the products from the previous step and as a first set of reactants, and add the same imported reactant step as a second reaction set, and we will have to adjust the configuration a bit. So for the reaction, again, we will have to use the same reaction that was used here. So I will, for now, copy paste the reaction specification from our previous node. We can check if we have the matching reaction conditions. Again, we can add here the same directional rule with the same tolerance value. And what is missing is, or what has to be adjusted is that we would like to react the first product molecule set from the previous node, and the second reactant set we are going to use the molecule column which comes from the mole importer node, and we still want to do a combinatorial transformation with a 1-to-1 ratio of reactants. And we can start the reaction. And hopefully it will finish in time. Again, I made some typos. By the way, if you have any questions now, or then feel free to ask them. Okay, hopefully I could fix the mistake here. Okay, and we have now desubstituted compounds as a result of the next step. However, if we check the unsuccessful reaction part, then we also find some combinations of reactants that could not be transformed. So I wanted to show you these steps in this example to demonstrate how the workflow can be extended, how the library generation can be extended and made more complete. Let's say what can follow now could be, for example, we can check if there are any duplicates among these as a result of this second reaction. We can filter them out, or if we want to, we can use additional reaction schemas besides these three or instead of these three. So there are still many points where the workflow can be extended. Also, the machine learning and the activity prediction part is something that can definitely be extended and made more accurate or more sophisticated, let's say. However, I hope that we could show you a workflow and an example that covers a large enough and interesting enough part of how drug discovery processes can be done with KNIME and ChemAxon.
Thanks, Dora. I guess it was pretty useful, and I hope that the other participants also feel so. So I guess we are pretty much out of time, but if anybody has any questions, probably we can take a couple of questions. I hope most of you are now able to import the workflow and run it. And if anybody has any questions, please let me know. I don't see any questions in the chat box also. So
No questions.
if there are no further.
Surya here, what about tomorrow? Like, what is that is pending tomorrow?
So we would be talking about the other workflow that Bilal mentioned, in which Bilal, can you just give a brief highlight about that?
So my workflow will be tomorrow about how to prepare data for a deep neural network machine learning model.
Okay.
Yeah.
Thank you.
Okay. So we will share the workflow and some details about the workflow today, just like we did yesterday. So at any point, if any of you have any questions or are facing any issues, please feel free to reach out to me. You can drop an email with the issue, and we will try to sort it out. And again, I repeat that the license that we have provided are for the nodes that are required for this workflows. If you want to further explore, please let us know. We will extend those licenses for additional modules, and these licenses are valid till 23rd of October. Okay. Thank you all. Have a nice evening. Thank you, Dora. Thank you, Bilal.
All right.
Bye-bye.
Bye.
Thank you.
Bye-bye. Thank you. Bye.